Hey everybody, welcome back to another video. In this video, we're going to talk about the intrinsic cardiac conduction pathway. It's going to be an interesting one. If you guys like the video, if you learned something from it, if you have any questions, please like, comment, subscribe, and let's get right into it. So, first things first, what do I mean when I say intrinsic cardiac conduction pathway? Okay, so you guys know how in skeletal muscle, okay, here's skeletal muscle, okay, the skeletal muscle requires a neuron, okay, a neuron to come and innervate it, okay, which will then excite this skeletal muscle and make it contract, okay. However, the heart muscle, it does not need that, okay? It does not need nervous innervation. It can have nervous innervation, but it doesn't need it. Okay, why? Well, it's because it has its own intrinsic conduction pathway, okay? Meaning that something inside of the heart is naturally just causing excitation of the actual muscle, okay? And that's what's happening. So, how is that the case? Well, in order to understand that, you have to know that the heart is made up of two main kinds of cells. There's other cells, but there's two main kinds of cells. 99% of it is going to be the contractile cells, okay? And if you remember from the last video, we called them the cardiomyocytes. Those are the ones that are going to actually be contracting and generating the power to pump blood. But then there's these other cells. These other cells are called autorhythmic cells. Autorhythmic cells. These autorhythmic cells, while they're small in number, they are the ones that are actually creating action potentials. Okay? And what do you guys know about action potentials? If you need a review, go back and watch my neuro video on this stuff. But action potentials are just a giant non-decremental wave of depolarization that will excite any excitable cell, like a neuron, like a muscle cell. Okay, so what do these autorhythmic cells do? Well, these autorhythmic cells, as we're about to see, they're embedded alongside the actual cardiac myocyte, the actual muscle, the contractile cell. They're embedded beside them. And they have the ability to create action potentials. And these action potentials, we know that all they are is just a wave of positive ions inside of the cell. And if you guys remember from the previous video, I said that these positive ions, okay, can flow from one cell to another in the heart through these channels called gap junctions, okay? Gap junctions. So when these autorhythmic cells generate an action potential, it'll start flowing everywhere, okay? But wouldn't that lead to something that's very uncoordinated? If all parts of the heart just started contracting randomly, that would be very uncoordinated. We couldn't pump blood out properly. So there is a specialized pathway that we have to follow in order to contract the heart properly. And there's three things that you must follow. The first rule that you got to follow is that the atria have to contract before the ventricle. Okay? Before the ventricle. And this should make sense to us, right? The atria have to contract and empty their blood out into the ventricles before the ventricle contracts. Because if the ventricle contracts first, well, guess what? It doesn't have any blood in it. Okay, so it just doesn't make sense. The second rule that you got to follow, the second rule is that the excitation of the cardiac muscle, okay, so the excitation. What do I mean by excitation? I just mean that the action potential has now reached the actual muscle fiber and is activating them to contract. So the excitation of cardiac muscle, okay, the cardiac muscle should be coordinated, should be coordinated, okay, in a way that each chamber of the heart contracts as a unit. Okay? But I'm not going to write the rest of that down, just this part. The excitation of the cardiac muscle should be coordinated so that each chamber contracts as a unit. The third rule that you have to follow is that the pair of the atria and the pair of the ventricles should functionally coordinate so that both of the pair, contract simultaneously. Okay? So both atria and ventricle should contract simultaneously. Okay? This means that I don't want just this atria contracting, ejecting its blood into this ventricle, and then this atria contracts, ejects its blood into the ventricle. No, I want it to be both of these contract at the same time, push blood in here, then both of these ventricles contract at the same time and push blood out. Okay? That's how the heart is beating. That's how the heart is working. These three rules have got to be followed if you want to live a good life, okay? If you want your heart to work properly. Now, what does all this have to do with anything that I mentioned beforehand, with the heart and the autorhythmic cells? Well, I'm glad you asked, because these autorhythmic cells have the ability to depolarize the heart or excite the heart muscle in a way that we want it to contract. Let's take a look. So the first autorhythmic cell that we got to talk about is the SA node. And the SA node is right here, okay? It sits right underneath the actual inferior vena cava in the right atrium, and it's only found in the right atrium, okay? The SA node is known as the pacemaker of the heart. Well, we'll come to learn that it's not the only pacemaker or autorhythmic cell, okay? But the SA node has the ability to generate action potentials. All right? We'll come back to this. The next thing in the pathway is called the AV node. Okay, the AV node. The AV node is another pacemaker cell. Okay, but we'll come to see that it does not actually set the pace for the heart. Okay, and again, we'll talk about this in a second. From the AV node, this special kind of bundle branches and branches and branches all the way to the intraventricular septum. And what you're noticing here is that the AV node is the only route of passing an electrical current to the ventricles. We'll touch on this a little later. But this bundle that comes down all the way here is called the bundle of His. Okay, it's pronounced His. Sometimes you might see this called the AV bundle. Now, I didn't tell you what AV and SA stand for. SA stands for the sinoatrial. So sinoatrial node, and AV stands for the atrioventricular node. So the atrioventricular node. Why atrioventricular? Because it sits right above the junction of the atria and the ventricle. Okay, you can see that. It sits right at the junction. The bundle of His is not an autorhythmic cell, okay? It's not an autorhythmic cell. It'll just conduct the action potential down. And again, I'll explain what I mean by this in a second. Let's bring this a little closer here. There you go. The next guy in this pathway is going to be this dude, the bundle branches. But specifically, before I write this down, you can see that the bundle of His will continue throughout the actual intraventricular septum, but separate into a left and a right branch. The left branch will supply the actual left myocardium. The right branch will supply the right myocardium. So we call this, for that reason, the left and the right bundle branches. Okay? Then you get these other things, okay? And let me do it like this. This guy right here. My goodness. You know what? I can't get it, but You see these little branches here? These little branches, all of these branches. Those guys are termed the Purkinje fibers. And these Purkinje fibers are little terminal branches of the bundle branch. Okay? They're little terminal branches of the bundle branch that actually go and squeeze their way in as close as possible to a lot of these cardiomyocytes that are just in here. Okay? Now, the Purkinje fibers, they actually exhibit pacemaker ability. They exhibit an autorhythmicity. Okay? So we've talked about three things that so far have a pacemaker ability. The SA node, the AV node, and the Purkinje fibers, they have pacemaker ability. But which of them is actually setting the pace for the heart? Okay, well, what do I mean when I say pacemaker? Pacemaker just refers to who sets the heart rate. Who sets the heart rate? And you guys know what the heart rate is, right? The heart rate is the amount of times the heart contracts in 1 minute. That's why it's measured in beats per minute. Okay? So the SA node, and all of these actually have a different kind of pace that they are able to set for the heart. The SA node, for example, sets an approximate 100 action potentials per minute. Okay, so guys, if it sets 100 action potentials per minute, okay, if the SA node generates 100 action potentials per minute, okay, what do you think the heart rate would be if the SA node was the pacemaker of the heart? Well, if you're saying 100 beats per minute, you'd be absolutely correct. Each action potential causes a single contraction, okay? Causes a single contraction. So 100 action potentials will call 100 beats, right? So we would have 100 beats per minute. Okay. What about the AV node? Well, the AV node has an approximate 25 to 40 action potentials per minute, okay? So if the AV node was actually the pacemaker for the heart rate would be somewhere around 25 to 40 beats per minute. The Purkinje fibers, having the lowest, they have approximately less than or equal to 20 action potentials per minute, okay? Per minute. Now, here's the thing. You might be thinking to yourself, well, what does this pathway even do? Like, what does it mean? Okay, let me explain something very briefly to you. This SA node, it will send signals. It'll generate, and actually, let me tell you this first. The SA node is the pacemaker of the heart. It sets the pace for every single one of these other guys, okay? Even though they have their own ability to become pacemakers, they won't be, because the SA node is at the top of the food chain here, and he is setting the actual rhythm of the heart, okay? So this guy, this AV node, wants to beat at 25 to 40, but the SA node says, nuh-uh, you're beating at 100, okay? I'm going to send action potentials to you at 100 per minute, and you're going to do the exact same thing to the rest of this pathway. So look, it'll send them this way, this way, this way, and it'll actually send this thin little connection here to the other atria. Okay, if you guys are curious, this little thin strand here is called the Bachmann's bundle, and it is just a interatrial connection between the SA node and the left atrium. What does that all mean? Well, the SA node causes a depolarizing wave or an excitation wave that depolarizes the atria first. So the atria will start to contract first, okay? They will start to contract first because the signal is generated here. The action potential is generated here. Then at the same time that signal reached the atria, all around the atria, that signal reaches the AV node. The AV node, though, is very smart. The AV node, remember I told you, is the only connection between the atria and the ventricle. That means if I sever the actual AV node from the ventricle, The ventricle's only way of contracting will become the Purkinje fibers. And obviously that's not viable because in that scenario, the SA node will still set the pace, which means that the atria are beating at 100, but then the Purkinje fibers are setting the pace for the ventricles at 20. So you can see the mismatch there. Anyways, the atria are receiving that signal from the SA node at the same time that, the AV node is receiving the signal from the SA node at the same time that the atria is about to contract. But the AV node is very smart. It actually holds on to that signal for about 0.1 seconds. We call this the AV nodal delay. Why does it hold on to the action potential signal? Why doesn't it just send it down the pathway to the bundle of His, down to the left and right bundle branches into the Purkinje fibers? Well, because this pathway is extremely fast. If it didn't hold on to this, if it did not hold on to this for 0.1 seconds, what would end up happening is the atria would contract at the same time as the ventricles. Or rather, the ventricles would end up contracting at the same time as the atria. And they would pump their blood all over the place. So, in order to avoid this from happening, the SA node depolarizes the atria first. The atria contracts. At the same time, the AV node receives that signal, but holds on to it for 0.1 seconds to allow the atria to contract. Then, after the atria have contracted, it'll send that signal down to the bundle of His, okay, down to the left and right bundle branches, and then all the way up the Purkinje fibers, which causes the ventricle to start contracting from the bottom up. The bottom up, okay? You see how the signal's coming on from here and then moving up? The ventricle is contracting bottom upwards, okay? All right. Imagine squeezing a ketchup bottle from the bottom of it, okay? That's how it's contracting. And the blood is moving up like that. Okay, so now we know about the AV nodal delay and why that's actually occurring. And now we know that the atria contracts first, squeezing their blood into the ventricles, and then the signal will move down over here all throughout, and then the actual ventricles will contract due to the signal reaching the Purkinje fibers, okay, and pushing their blood out. And then the cycle will restart all over again, okay? So that's how that stuff works. Now, this right here is the reason why the heart can actually beat on its own. This intrinsic cardiac contraction pathway, conduction pathway, okay? And it's because of these pacemaker ability or the autorhythmic ability of this SA node cell. If you were to damage the SA node so that it doesn't work, well, the next pacemaker that can take the job is the AV node. If you were to damage the AV node, the next pacemakers would be the Purkinje fibers, okay? Awesome. Great. But again, you don't want that because, the Purkinje fibers are not beating or don't have a high action potential rate. And they're only actually connected to the actual ventricles and not the atria. Okay, so you don't want that. You want this entire pathway to work properly. All right, starting at the SA node, which sets the pace for every single one of these guys. Okay, even though the Purkinje or the AV want to beat at like 25 or 20, they're not going to because the SA node is going to set the pace. Now, what do you guys know about your actual heart rate though? So I said the heart rate of the SA node is active, right, is about 100. But why isn't your heart rate at 100 right now? What is your heart rate normally? It's around 60 to 70 beats per minute. Why? Well, this is because your heart doesn't beat independently of the nervous system. It can because of this pathway, but it doesn't. It also has nervous innervation, okay? So the sympathetic nervous system and the parasympathetic nervous system play a role on its contraction, okay, or its heart rate abilities. Okay, what do I mean here? When you're at rest, your heart rate is 60 beats per minute. This indicates that your heart rate is actively being lowered because if the nervous system wasn't interfering, your heart would want to beat at 100. But A part of the nervous system is actually actively telling your heart, hey, relax, calm down. And at rest, the nervous system that is doing that is called the parasympathetic nervous system. Remember that? It's called the rest and digest system. So at rest, you don't need your heart rate to be high. Why would you? So what you would do is you would actively suppress it. You would bring it down to 60 beats per minute. But let's say a bear walks in, your sympathetic nervous system, your fight and flight kicks in and actively ramps up your heart rate. Okay? Awesome. Okay, now we can actually take a look at this. Okay, so now we can take the SA node. Okay, we can take the SA node and look at how it is actually generating that pacemaker. Okay, how does it actually spontaneously generate this pacemaker ability without the need of any sympathetic or parasympathetic activity? How? How does it just come up with this randomly to depolarize all of these contractile cells? But one thing I do want to mention is a lot of people get confused is all of these pathways, these are all embedded within the actual muscle, okay? They're all intertwined between the muscle, okay, between the cardiac myocytes. And they also have gap junctions connecting them to a cardiac myocyte, okay? So let's say this right here is an SA node cell, okay? And this right here is a cardiomyocyte, okay? And this right here is the actual AV node, okay? AV node cell, right here. There is gap junctions between this guy, there's gap junctions between that guy, okay? So you can see how, and this is a rather simplified view of it, but there's gap junctions between all of these guys, okay? So when the SA node fires, you can see what I mean, where the cardiomyocytes in the atria will contract, the AV node will receive it, hang on to that signal, pass it on to the bundle of His and the left and right bundle branches and the Purkinje fibers, which are, again, embedding themselves, those terminal branches, they're embedding themselves in between the actual cardiac myocytes, touching as many of them as they can to cause this synchronous contraction between both of the ventricles, okay? So that's how we synchronize contraction as well, through this pathway, okay? And again, through this pathway, we meet all three of the things that we said we want to meet at the start of this video. Okay, now let's look at how does this actual SA nodal cell generate this pacemaker potential, okay? How does it have that ability? We actually call it autorhythmicity or automaticity. How does it have that ability? Okay, well, in order to do that, you have to remember a little bit about your action potential graphs, okay? And remember that, there was a portion of the graph called the threshold potential, there was a portion of the graph called the membrane potential, and there was a portion of the graph that was the peak part of depolarization. Just a brief reminder, once you are making the cell more positive, that's called depolarization. When you're making the cell go back to its resting membrane potential, it's repolarization, and making it negative is hyperpolarization. Okay, so in any excitable cell, I want to remind you that in any excitable cell, the concentration gradient of ions looks like this. An excitable cell just means like muscle cells or neurons. Potassium would be extremely high, and potassium would be extremely high inside of the cell, but it would be low outside of the cell, okay? Which means that if you let it, it would want to go out of the cell, which would make the inside negative, right? Because a positive charge leaving the inside of the cell would make the inside negative or less positive. A positive charge like sodium, for example, is high outside of the cell, but low inside of the cell. So it would want to come down its concentration gradient, okay? Other things like calcium are low inside of the cell, but are high outside of the cell. And again, they would want to come down their concentration gradients, okay? So this is what the concentration gradients of these ions looks like in any excitable cell, okay? The bigger it is, it means it's higher. And the smaller it is, it means it's lower, okay? So potassium higher inside than outside. Sodium higher outside than inside. Calcium higher outside than inside. Okay, cool. That's going to be important when we look at the mechanism. One thing you have to know about these SA nodal cells is that they don't really have a stable resting membrane potential. So, you know how in a neuron it's about negative 70 millivolts? I can't actually sit here and say that the resting membrane potential of an SA nodal cell is negative whatever. They don't have it because their resting membrane potential really doesn't exist. Their membrane potential is constantly fluctuating. And that's because of the fact that they are pacemakers. They're spontaneously just depolarizing themselves. So there is one point, though, where we can say that the depolarization kind of starts. It starts around this negative 60, okay? And then it reaches a peak of 0, okay? 0 millivolts. And, depending on where you read, it might be different. The most up-to-date text that I have says it goes up to about 0, maybe sometimes positive 10. Okay? But it might differ depending on where it goes up to. Now, this is the SA nodal action potential. But before I tell you about the action potential, let's take a look at how does it actually look like at the cellular level. Okay? So at the cellular level, you have this red channel here. This red channel is a potassium channel. It's a potassium-gated channel, voltage-gated channel. And at the start of this scenario, at the start of this red line here, okay? These are all color-coded. At the start of that red line, the permeability of potassium is decreasing. What do I mean by that? What does permeability mean? If you guys remember, permeability talks about the ability of something to pass through the actual membrane of the cell, okay? So here is one SA nodal cell. Here is the intracellular fluid. Here's the extracellular fluid, so outside of the cell. And if I say that potassium's permeability is actually going down, it means that it's no longer able to cross the cell and go outside. Okay? Why does it want to go outside? Because of its concentration gradient. Why is its permeability decreasing? Because this potassium voltage-gated channel is closed. So at the start of this scenario, potassium tries to escape, but it can't. Okay? But it can't. And that's going to have some implications. That's going to raise the membrane potential a little bit. Okay? Now, another thing is that at the same time this is happening, at the same time that this right here is happening, all right, the changes in the membrane potential are sensed by, and let's make this a bit bigger. Let's go up to 6 here, and boom. The changes in the membrane potential are sensed by these special sodium channels. They actually have a funny name. They're called the funny or F-type sodium channels. Okay, sodium channel. The F stands for funny type sodium channels. And you don't have to know why they're called funny. If you want, is because they behave in a manner that is different than other sodium voltage-gated channels. Okay, so usually with sodium voltage-gated channels, they open when a cell is depolarized, but these guys, they start opening up when the cell is a little bit hyperpolarized. So that's why they're called funny, but you don't have to know that. Now, when these guys open, they like to bring in sodium. Okay, you see that? The voltage-gated channel, it's opened now. The gate is open. Sodium will come in, and it'll make the inside of the cell positive. Okay? So remember how we said we start around negative 60? Well, we start around negative 60, and the sodium channels open up. Okay? So negative 60, once the sodium channels open up, maybe we're around negative 55 millivolts now. Okay? And this sodium will come in and make the membrane depolarized to around negative 55. Okay, just a hypothetical number. And what's going to end up happening is this sodium will come and open, or these positive charges rather, will open up this voltage-gated calcium channel. A specific kind of voltage-gated calcium channel, though, not just any. It's called a T-type voltage-gated calcium channel. Okay, and it lets in the ion called calcium. Okay, so here's calcium coming in, and boom, it makes the cell very, very positive. Look at that. Okay, so initially it was these 3 plus signs. Now we have these 6 positive signs. Okay, so it makes the cell very positive. Now, something I have to tell you. Okay, remember I said that this is color coded with the graph. So let's go with that for a second. First things first, this red portion. The permeability of the cell to potassium decreases. Okay, decreases. You guys know how potassium likes to leak out of the cell, right? Move out of the cell. So if the channel was open, then this line, rather than going back up, it would actually continue to go down because it would make the cell more negative if potassium kept leaving. But you see that this line here starts to kind of increase. And that's because this gate closes and potassium no longer can leave out of the cell. And if that's the case, it stays in the cell and makes the cell a little bit more positive. At the same time that was happening, these funny sodium channels, okay, these funny sodium channels, they actually opened up. Okay, they opened up their gate, their voltage gate. Okay, and then when they opened up, they brought in some sodium. And look, they actually made the membrane a little bit more positive. Okay, you see that? We're a little bit more positive now. When they made the membrane more positive, they open up another special kind of channel called a T-type calcium channel. Do you guys know what T stands for here? The T stands for transient. You guys know what transient means? It means short term, okay? That means these channels are open for a short period of time. Okay, these transient channels, they bring in calcium, okay, after being opened up by the actual positive charges from the sodium. They bring in a whole bunch of calcium, depolarizing a cell until they reach threshold potential. Okay, what is threshold potential? If you guys remember from my neuro video, I mentioned that the threshold potential is the voltage required to launch an action potential. You have to hit this voltage to hit an action potential. If you don't, then you're not launching an action potential. Okay, so that means that the T-type channel brings in enough calcium to bring us to the threshold potential. All right, but you know what's happening to these channels? After this sodium channel brings in enough positive charges to open up the T-type calcium channel, guess what happens to it? It will then close its gate, okay? Meaning it will no longer be able to bring in that sodium, okay? Same thing with this T-type calcium channel. After it brings in enough calcium for us to hit the actual threshold potential, it too will close its gate and become closed. But what happens to those positive charges, right? So remember, now we came from negative 55 to negative 40 millivolts. That's threshold potential. At threshold potential, guys, a special kind of ion channel opens up. Voltage-gated ion channel opens up. And that one is going to be this other type of calcium channel. It's called the L-type. The L-type calcium channel. Okay? L-type calcium channel. The L-type calcium channel, the L specifically stands for long lasting, meaning it's open for a long period of time. And you can see that this guy's a calcium channel, so it's going to bring in some calcium, okay? Boom, and this guy is going to bring in a whole bunch of calcium, and it's going to depolarize the cell a lot, guys, okay? And it's going to depolarize the cell to about 0 millivolts, okay? 0 millivolts. You can see that right here. So from this negative 40, which was brought by the T-type calcium channels, we open up the actual L-type calcium channels at that voltage, the negative 40, and then these positive charges from the L-type calcium channel flow in all the way to around 0 millivolts. At 0 millivolts, this channel will then close, okay? And another channel opens up. Another channel opens up. What kind of channel is that, guys? What kind of channel is that? That channel would be the potassium channel that we saw in the beginning, guys. Remember that one? that potassium channel will open up at this 0 millivolt point. And guess what it's doing? It's going to let potassium out of the cell, right? Down its concentration gradient. And if potassium goes out of the cell, what happens to the membrane potential? It becomes negative. Potassium will keep going until we reach that negative 60. Okay? Look at that. So here's the potassium. Okay? The gate of the voltage gated channel is open now, and potassium is going to leave and make the inside of the cell negative. Okay? And it'll do this until what? Until it closes. Which means what? Which means the permeability of the potassium will then drop. Okay? And then the cycle repeats again. Do you see the cyclical pattern here? Do you see how it's a cycle? This is exactly how the SA node is able to keep spontaneously generating these action potentials, and it has this pacemaker ability. Okay, there is a special portion of this graph that I have to talk about. From here all the way to here, that point right there, we call that a slow depolarization. Why slow? Because usually when you have depolarization, such as this, it's pretty fast. It's occurring very fast, actually. Okay? But in this scenario, it's actually very, very slow. You can see that it's taking its time to depolarize, right? And it's using multiple different ions like sodium and the T-type calcium, okay? And this also has another name. So on top of being called slow depolarization, it's also the pacemaker potential. So this right here is the pacemaker potential, and this portion is just the action potential. Okay, that's just the action potential. All right? Okay, cool. Now that you know this, okay, I can actually switch to the color that I originally drew this in, and it's this, darkish purple color, and we're gonna say that that color right now represents the normal SA node pacemaker, okay? Pacemaker. And you'll see why I'm doing this in a second, okay? But now you guys are familiar with why this actual SA node is able to depolarize itself. Okay. But remember, I said that the actual nervous system can also modulate the activity of the heart rate, okay? And who was the heart rate set by originally? The SA node. So if that's the scenario, that means that these guys must be acting on the SA node. The sympathetic and the parasympathetic nervous system must be acting on the SA node to modulate the activity. And yes, they are. So here's the brain. And from the brain, we can show right here a neuron, and here another neuron, okay? And in red is going to be the parasympathetic nervous system neuron, and in blue is going to be the sympathetic nervous system neuron, okay? And we're going to take a look over here on these graphs, how they're actually changing the shape of the graph, okay? What are they changing about the graph? Okay, so first, let's look at the parasympathetic nervous system. And I want you guys to tell me, after I draw this, what is the change? What is the difference that you're seeing, okay? All right, so here we go. Let me draw this, and then let's draw it going all the way up and down again. Okay, so what do you notice about the SA node, okay? The SA node, but this time with the parasympathetic nervous system action. Okay, so now the parasympathetic nervous system is acting on the SA node. What happened to it? Did the peak of depolarization change? No. Did the shape of the actual action potential change? No. So what changed? What changed was this right here, the pacemaker potential. The pacemaker potential, or the slope of the actual slow depolarization. That's what changed, guys. Pay attention. This was the original, okay? In this purplish color, that's now a little faded, that was the original. So this right here is the original SA node. And you can see that in this time period, okay, some arbitrary time, we actually did 1 and 2 action potentials. But in this same period of time, with the SA node action with parasympathetic activity, we actually only launched one action potential. Okay, so normally we did 2, but with the parasympathetic, we did 1. What does this tell you? This tells you that the parasympathetic nervous system is slowing down the rate of my slow depolarization, which means it's slowing down the rate at which I reach the threshold. If it's doing that, that means it's taking me a longer time to launch the action potential. And because of that, I only have 1 here rather than 2. Okay? And it'll keep doing this, right? This slow kind of very long depolarization until my heart rate is down, okay? So with the SA node, originally my heart rate is about 100 action potentials or beats per minute. But with the SA node and parasympathetic nervous system, let's say it drops down to about 60 beats per minute, okay? Cool. Now let's look at the sympathetic nervous system. And I want you to tell me what is different there. What is different with the sympathetic graph? So in the sympathetic graph, we're going to start out right here again. Okay, so what do you notice here? What do you notice here? Okay, look at this. Again, did the shape of the actual action potential change? No. Did the maximum depolarization change? No. And if it did, it's just my terrible drawing. But what did change? Again, it's the slope of that slow depolarization or the pacemaker potential. Okay, the more steep it is, the faster I reach the threshold potential and the more action potentials I can generate. Okay, the less steep it is, like this, that means the less action potentials I can generate because the longer it takes for me to actually reach the threshold potential. Okay? All right. And you can see that. You can see that right over here. I actually generate 1, 2, 3 action potentials, whereas with my regular SA node, I would generate 1 and 2. So this right here is your SA node with sympathetic nervous system activity, and let's say it raises your heart rate to about 120 beats per minute. Okay? Now you guys can see how we can modulate the SA node activity with the sympathetic and parasympathetic activity. And now you can also appreciate the importance for the slope of this pacemaker potential. Okay? If the slope is very steep, I reach threshold faster and I launch more action potential. If it's not so steep, I reach it later and I launch fewer action potentials. If it's normal, then I'm launching the normal amount. Okay, guys, that's going to finish it for this video, okay? We talked about the intrinsic cardiac conduction pathway and how the SA node is able to spontaneously generate this pacemaker potential. I hope it made sense to you. If it did, if it really truly helped you, please like, comment, and subscribe. It really helps the channel. And I will see you in the next video.